Good afternoon, everyone. Dr. Fernando sends his apology for not being able to be here because he's not feeling very well. I'm Hui Ng from the University of Queensland. I work closely with Dr. Fernando in this project in the group, and I'll be presenting on his behalf. So today I'll be here to try to convince you that the nanopatch is the best way to do vaccination. So let me first introduce you what the nanopatch is. So the nanopatch is a 4x4 silicon microarray. It contains, doesn't really work. Okay, well, it contains many small micro projections, and it's etched using deep reactive ion etching microfabrication. So it contains 3,000 over small micro projections. So this is the mouse prototype. The length of it is about 100 microns long, and the density, it's a highly dense projection microarray. So since the birth of the needle and syringe, we have been using the wrong method for vaccination, and I'll present to you some data that prove my theory. So the current method for vaccination targets the skin, so IM delivery, which has the least amount of immune cells, and therefore you need a lot of vaccine to get a seroconversion or protection. And then there is also the intradermal injection, which targets the right area, which is the skin. That's a lot of immune cells, but there's a need for skilled personnel like the doctors and nurses to perform this delivery. It's still a good improvement, but there's still needs for refrigeration for these two methods for refrigeration for liquid vaccines. And then comes the microneedle, which is very similar to the nanopatch, but it's a lot bigger, a lot longer, a lot wider, and it's less densely packed. And this makes the nanopatch much unique. So by the naked eye, you can't really see any projections on the microarrays, as you can see over here in the picture. So this is a cryo-SEM of the nanopatch immunized into the skin. So it's force-colored to differentiate the different layers of the skin. And you can see that the microprojection actually penetrates through the skin and delivers the vaccine. So there's so many advantages of the nanopatch, and it is needle-free, so it removes all risks that can arise with the needle, like the needle and syringe, like needle stick injury in the hospital, and also needle phobia that could potentially reduce patient compliance for vaccination. It can potentially be pain-free as well, because nanopatch only interacts with the top layer of the skin. It reduces vaccine dose, so I'll be presenting you some data later. Possibility of self-administration, and I'll also show you pictures of how the nanopatch can be applied to the skin in animal models. We have also shown stability in room temperature because we dry coat the vaccine onto the patch. Logistically, the nanopatch is really small and can be stored in room temperature, making the transportation a lot easier and a lot more convenient, and definitely ideal for pandemic situations. So this is the SEM of the projections as well as the beaver of a 29 gauge needle. So you can see at the background there's many small projections that's on the nanopatch as compared to the foreground, which is the beaver of the 29 gauge needle. You can compare how big the beaver is as compared to the microprojections. So we showed that we can coat the patch with the vaccine. So the panel A and C is before coating, B and D after coating at different magnifications. So we have shown that we can coat the patch, the tip of the patch, and we have shown also to be vaccines to be stable at room temperature for up to 6 months. So after coating the vaccine, you want to know how to apply the patch. So this is an illustration of how we apply the patch with a spring-loaded applicator. It is actually very straightforward. So for the mouse model, we anesthetize the mouse, place the skin just right below the square platform underneath the applicator, and just by the pull of the pin, you could actually apply the nanopatch. So anyone in the room here can do it. And this is how the patch ear skin looks like. So after application, you can see the projections on the skin straight after. And with a dye, we can actually see the trail of the microprojection that penetrates through the skin. So it's only just the top layer, the viable epidermis, and the top layer of the dermis. So this is an image captured by NPM. So the red indicates the vaccine. The green indicates MHC class 2 cells. So most of the immune cells contains this marker. So you can see how the vaccine is actually delivered into the close vicinity of those immune cells. And over here you can see actually how the cells is actually interacting with the vaccine, really closely packed together. So how does the immune response looks like with the nanopatch as compared to the traditional way? So in the animal model, we have shown that at low doses of nanopatch, we induce similar antibody response. So on the left side, left box is the IM injection, middle box is the nanopatch injection, and then the right side is just the negative control. So at 34 nanograms, you can see that we induce equivalent antibody response at 6,000 nanograms via the IM injection. So the top panel is 21 days post-delivery, and the bottom panel is 63 days post-delivery. So long-term and short-term immune response. And if you're not familiar with influenza vaccine, so FDA have a guideline for influenza vaccine efficiency. So if you get HI titer of more than 40, you are this vaccine is considered protective. So the animal models that we used are all naives, so you shouldn't see any response at all. But after immunization, you can see that at all doses we induce HI titer, and above 34 nanograms, we all have protection. But for IM delivery, even at 6 micrograms, 6,000 nanograms, the second strain over here only really induced on the mark 40 HI titers. So not only antibody response and HI titer, but we also shown that we also induce cellular response. So cellular response are much better with the nanopatch as compared to IM. We showed that at 5 micrograms immunized by the nanopatch, we induce similar CD8 T cell response as compared to IM delivery at 500 micrograms. So that's about 100 times dose reduction. So here I've shown so much advantage of the nanopatch. We consistently show that at low dose we induce similar response as the IM at higher dose. Nanopatch is designed to target immune cells in the skin. It's potentially pain-free, and we could also deliver it, self-apply nanopatch ourselves. So it has a great potential to replace the needle and syringe. Well, then again, why is this happening? Because we are only targeting the skin. Intradermal delivery also targets the skin, but it doesn't show as higher immune response as the nanopatch. So we've just published in JID showing that there's possible sterile inflammation, and this is what we hypothesized could be happening. So here we compare intradermal delivery on the right side, nanopatch delivery, and untreated skin. So this is an NPM image. Green depicts the live cells, red depicts, or magenta depicts the dead cells. So after application, immediately after application, you could see the dead cells around the projection where the nanopatch is applied. So when we apply the nanopatch, we create a layer of controlled cell death around the projections, and this might have an effect that increases the immune response. And we term this the physical immune enhancer. So comparing to ID, the nanopatch is achieved a lot, there are more cell death achieved, as you can see on the graph on the side. So nanopatch achieved roughly about 20% cell death, but ID less than 10. So maybe this is the way to go. More cell death can be better. Well, by creating more physical inflammation, we did not see an increase of antibody response. So the first panel here is a reduction of projection, so less projection, less cell death. The middle panel is our standard application, and then the right panel is a multiple application on the same spot. Try to double up the amount of cell death. But the antibody response started to decrease when we double up the number of the amount of cell death. And you can see the graph on the left, on the top right side, where we have ID having like less than 10% of cell death. A reduction of microprojections have about 10% of cell death, and then our usual application has about 20% of cell death, and doubling up by 3 times the immunization, we create double the amount of cell death. But if you can see just right at here, which is the antibody response, the antibody response starts to increase, and then when the nanopatch, well, when we increase the number of cell death, antibody response starts to decrease. So there's a sweet spot of how much cell death can increase immune response, and the nanopatch has already achieved with just one immunization. So more cell death doesn't mean it's better. There's always a threshold for it. So we're still curious, how does it work? So now with the full mouse genome being mapped in December 2002, we have the technology to screen all the different genes in the mouse and determine what is actually regulated. So using RNA sequencing, we are able to capture the genetic profile and determine the gene regulated by the nanopatch when we compare it to the ID. So after a long fight with bioinformatics and statistics, we are able to narrow down from the 22,000 genes that is present in the mouse genome. We managed to narrow it down to just a couple of thousand genes that is significantly regulated. So here's the heat map of the different cytokines and chemokines. So what I did here is ID at 1, 4, and 16 hours post immunization, and nanopatch at 1, 4, and 16 hours post immunization. So we're able to capture many different regulations of genes. Sorry, nanopatch highly regulates many immune cytokines and chemokines, as you can see from here, where there's more red and there's more blue for ID. And ID only regulates those immune cells at a later time point, which is this, just the last row here. So there are many pro-inflammatory genes such as IL-1 beta that we have seen here that's highly regulated at all the time points that the nanopatch has induced. Or there is at least another hundreds or thousands of genes that are being regulated, and some of them have known function, but there are a handful of them that has no known functions yet. There could be any bystander genes that could be responsible for just metabolic processes and can have no function in the immune response. So there are still a lot of work to be done, and RNA sequencing just gives us a broad insight of the gene regulation of the nanopatch as compared to the ID. There are a lot of data mining and correlation studies to do as well. Genetic response gives us a good idea of what is going on, but we still need to do work to confirm the translation of the genes into proteins and ultimately the function of the protein as well. So I would like to thank all the funding body, Professor Mark Kendall and Dr. Fernando. This is supposed to be his talk, and everybody in the research group. Thank you very much, and I'll take questions.
Yes, question?
Yeah. What's your sample for the RNA extraction? So blood, or what sample did you use for the RNA?
Skin sample. So we are interested in the localized response of ID and
I noticed as well that the IL-10 response was obliterated, so that's anti-inflammatory. Obviously, so that maybe also is significant.
Yeah, there's a lot of things that we wanted to look at. So yeah, we're still Thank you very much. Yeah.
How
how do we formulate so the formulation includes a viscosity enhancer, metacellular. So we published this some time ago as well. So viscosity enhancer, the vaccine itself. So the vaccine will be a licensed vaccine that has been used via intradermal. Yes, that's right. And then we dry coat onto the patch. That's right.
Any other questions? About 8 years ago, Barbara Fazekas in Sydney at the Centenary Institute published a paper showing that if you target antigen to Langerhans cells in the epidermis, you actually tolerise the immune response. Have you seen any evidence of that? Because it's a paper that's often quoted to present a case against skin vaccination, but I don't believe it myself. But have you had any evidence of that?
They are controversial between whether you want to target the epidermis or you want to target the dermis. So the epidermis contains Langerhans cells, which you say tolerize, and targeting the dermis having dendritic cells, dermal dendritic cells, is where we think it's giving us the immune response. Well, then again, there are people that believe that Langerhans cell also gives a good immune response, a pro-inflammatory response. So I would think that it depends on what you inject into the skin and maybe the size of the formulation, the size of the vaccine or the peptide that you're putting in. It will tweak it from Th1 to Th2 or tweak it from tolerance to immune response. So far we have only looked at model antigen, which is very purified. So I cannot comment on that yet, but we are looking into that.
Okay, if there are no further questions, thank you very much.
Thank you very much.